Okay, hello, this is going to be Chapter 4, Part 1, Tissue Level of Organization. And introduction here, and we're going to talk about tissues, is the cellular organization level intermediate between cells and complete organisms. A tissue is an ensemble of cells, not necessarily identical, but of the same origin. And then next term here we're going to learn about is histology. It's the study of the microscopic anatomy of cells and the tissue of plants and animals. So whenever we talk about tissues, the organs have to be made of something. So even if we're talking about the heart, it has different types of tissue in it. It's going to have areas of ligaments that hold it, vessels that come into it so that it can pump the blood, and then the actual heart tissue itself, or the muscular tissue. It'll have various surroundings, pericardial sac being an example of this. So the tissue here is the cellular organization. So one of these cells would be known as a heart cell. Well, you get a million of them together, and now you have a tissue. And this is figure 4.1 in your books, an orientation of the tissues to the body. So first thing we need to look at here is we have cells. And the cells have a specific job. They're either going to secrete and regulate extracellular material and fluids. So this is how that specific type of tissue is going to interact with the environment. And these cells pretty much combine and they form tissues. An example of this is going to be the heart tissue again. And in these tissues we have all different kinds. So we have epithelia, which are going to cover the exposed surfaces. So this is going to be the outside. We have connective tissue, fill internal spaces, provide structural support, and store energy. And then we're going to have muscle tissue that's in that. This would be more specific. This is a type of muscle tissue. Contracts to produce active movement. And then we'll have neurotissue in these tissues. And those tissues combine to form organs. The example of this, if we're talking about the neural tissue would combine to form the central nervous system. And then these organs will interact with the organ systems. And we're going to start covering these organ systems from chapter 5.1. So we'll go through them each by chapter and then kind of continue on until we get all the organ systems covered. Epithelial tissue. We're going to talk about the functions of the epithelia, intercellular connections, epithelial surface, basement membranes, epithelial renewal and repair, how it kind of repairs itself, classifying epithelia, and then glandular epithelia. In the epithelial tissue, this includes epithelial tissue and glands. So all the glands that we have throughout our body and all of the surface tissue is considered all epithelial tissue. And then another term we have here is epithelia, a layer of cells that cover internal or external surfaces. So an example of this, your skin is epithelia. So this kind of covers the entire organ system. It's considered its own system called the integument. Glands, composed of secreting cells that are derived from epithelia. An example of a gland is endocrine glands are all throughout your body, and they secrete things that allow the organs to kind of communicate cohesively. And then avascular. In most epithelial on the surface, it has the absence of blood vessels. Avascular epithelium. Okay, characteristics of epithelial cells. The cells are closely bound together. A free surface exposed to the environment, attachment to an underlying connective tissue by a basement membrane, the absence of blood vessels, and epithelial cells that are damaged are continuously replaced. So on the surface of the skin, what happens is we will have tons of cells underneath it, and then at the lower structures, we'll actually have vessels that are in there. So if you get a laceration or a cut, and you cut into it and it bleeds, you've cut into it deep enough to where it's actually in that vascular supply. Functions of the epithelial. One, to provide a physical protection. So we can't keep the organs pretty much exposed to the environment. They protect exposed and internal surfaces from abrasion, dehydration, destruction by chemical or biological agents. Example, if you got strep or staph on your skin, a good chance that the skin would protect your organs underneath from that infection, and it could be washed off. If it found a way inside of there, then it would probably get infected at a later point. 2, they control permeability. Any substance that enters or leaves the body crosses an epithelium of some sort. So this is all the mucous membranes are considered epithelia. All the lung tissue are type of epithelia. Intestinal tract type of epithelia. Provides sensation is the next one. Detects changes and relays to the nervous system. So you have all the sensory input that's in the actual epithelia. Let's kind of mark these off here. And then produce specialized secretions, which are the gland cells. We either have 2 types of gland cells here. We have exocrine and endocrine. An exocrine gland, secretions are discharged onto the surface of the epithelium. So this would be like sweat glands and things like that. An endocrine gland, secretions are released into the surrounding tissues, fluid and blood. Secretions are also called hormones. So an example of this, you have adrenaline glands. So the adrenaline glands are an endocrine gland. That's because they release hormones that talk to the entire body and keep it cohesive or ask it to do specific functions. Example of this would be fight or flight. You release epinephrine or norepinephrine or adrenaline from your endocrine glands, your adrenaline gland. So those hormones would go throughout the body and kind of cascade an event or a fight or flight response. Intracellular connections. We have 3 types that we're going to talk about here, which are tight junctions, gap junctions, and desmosomes. A tight junction is a lipid layer of adjacent membranes, and they're tightly bound together by interlocking membrane proteins, and we'll see these here in just a second. Gap junctions, 2 cells are held together by embedded membrane proteins, and these are kind of like sucker rods that are going down through cement, and I'll show you what those are. They kind of anchor the cellular environment together. And then we have a desmosome. A cell membrane of 2 cells are locked together by intercellular cement and by membrane proteins connected to a network of intermediate filaments. And these are kind of a little bit of a bigger type of intercellular connection here. And here we go when we're looking at these. This right here is a tight junction, and it's just pretty much these interlocking membrane proteins here have cemented these membranes together. This is an adhesion belt, and if you'll notice here that they kind of have protein anchors that are into the actual substance or the other intermediate filaments in the other part of the cell. This one here is a gap junction, and this would allow for collateral communication between 2 cellular bodies. Here we have a button desmosome, and that's down here, right here. And if we'll note, this is anchored pretty good together by cellular adhesion molecules and intercellular cement, so to speak, that sticks it together. And you have all of these intermediate filaments that are running through there and kind of anchoring it like sucker rods. A hemidesmosome, which is down at the bottom here, is even more anchored. These are kind of like J-bolts that you would put in a house. So these intermediate filaments that give the cellular environment structure, these things are going to wrap around that and then be in the basement membrane or the cellular cement, so to speak. So types of intracellular connections that keep cells together. The epithelial surface. The apical surface of the epithelial cells often have specialized structures that distinguish them from other body cells. And the 2 types we have here are microvilli and ciliated epithelium. The cells that have microvilli add surface area approximately 20 times to that of a non-villide cell. So I think I told this in the last lecture that we did when we were talking about surface space. So if we have a flat surface space, we don't get near as much as if this thing has all these nooks and crannies. And all of these little microvilli here, if we stretch this out where it was flat, it would be a much larger area. And what those microvilli do is allow us to have tons and tons and tons of surface area for things to be exchanged. Examples of these are going to be in the small intestine and in the lungs. Ciliated epithelium moves material across the epithelia, and the example of this, these are ciliated, these have little hair-like projections or finger-like projections. It's actually in the tissue. And these are in the example of this is going to be in the respiratory tract. And here they are. And microvilli are these little tiny ones, and the ciliated ones are a little bit larger. Take a look there. And if you can see them here, this is a blown up picture. This is the microvilli, and then these are the cilia. And then this are the cells underneath. Golgi apparatus, which would be providing secretions, the nucleus, and then energy production from the mitochondria. Basement membrane. 1.8, the basement membrane. Basement membrane that consists of protein fibers firmly attached to hemidesmosomes, and a hemidesmosome is kind of a half. It's the one that we looked at previously that had the J bolts in it, and I got another picture of this coming up. And this down here is a hemidesmosome, so this thing is anchored pretty good. It's got cellular adhesion molecules in here that are hooking onto this inner filament and anchoring into the basement membrane. So hemidesmosome, pretty strong anchor as far as an intercellular connection goes. Epithelial renewal and repair. Stem cells. And a stem cell is pretty much a blank cell before we get too much into this. They are biological cells found in all multicellular organisms. They can divide through mitosis or cellular separation, what we learned in the last chapter, and differentiate into diverse specialized cell types, and can self-renew to produce more stem cells. So what you generally have is at the bottom of these things, you have these blank cells. So in case, this one's a little bit more, these are actually, let's say that these are epithelial cell types, cuboidal. So as the cell dies off or gets injured, this cell here will replace it. So then it becomes more specialized and becomes this type of cell. But a stem cell, whenever it starts off, is pretty much a blank cell. This is where we get things like stem cell research. And the big push on stem cell research is to get them to look into stem cells in spinal cord injuries. If you sever a section of your spinal column, the spinal column grows back. However, it grows back at such a slow rate that the complications of your injury will probably kill you before you grow new spinal tissue. So the decreasing that rehab time is why stem cell research is so important in this. That an infusion of stem cells would occur into this blank area, and they would receive the information from the more specialized cells above and below and become neural tissue. Classifying epithelia. Two types of layering. They're simple and they're stratified. Simple consists of a single layer of cells that cover the basement membrane. Simple epithelia are characteristic of regions where secretions or absorption occurs. And then we have stratified. Provides a greater degree of protection because there are several layers of cells above the basement membrane. Stratified epithelia are usually found in areas subject to mechanical or chemical structures, such as the surface of the skin, linings of the mouth, and anus. Three types of cells that we're going to look at in this classification: squamous, cuboidal, and columnar. As squamous, the cells are the thinnest. These things kind of look like an egg as you're looking at them. They're thin and flat, and the nucleus occupies the thickest portion of each cell. Then we have cuboidal, who resemble little hexagonal boxes when seen in 3 dimensions, but in typical sections view they appear to be square. And we'll see these in the next slide. And then columnar. The cells also have hexagonal, but taller and more slender. The nuclei are crowded into a narrow band close to the basement membrane. And this is our simple. So remember, simple are 1 cell layer thick. And we have squamous, which kind of looks like an egg. Then we have cuboidal, which are kind of squared off. They look like a cube. And then we have columnar, which look like columns. Looks kind of like Bart Simpson up here. So simple epithelia, they're going to be 1 cell layer thick. Stratified are multiple cell layers thick. So these are the stratified squamous. Again, they look like an egg. These are stratified cuboidal, which are still kind of cube, but they're kind of stacked up. And then these are stratified columnar. They look like a column. So squamous, cuboidal, columnar. Simple means that they have 1 cell layer thick. Stratified meaning they have multiple cell layers. So examples here of simple epithelia, and this is figure 4.4 in your book. Simple squamous epithelia, lining of the peritoneal cavity. These things are going to be very, very kind of looking like an egg here. And this is a real picture of them. Oh, sorry about that. This is a real picture of them here on this side. And then we have simple cuboidal epithelium, which this again is one cell layer thick. Example of this is going to be in the kidneys or the kidney tubules and the ducts, cuboidal cells. This is where water maneuvers. You have a glomerulus, proximal, loop of Henle, distal tubule. So how your body forms urine moves through these cells called simple cuboidal epithelium. And then we have simple columnar epithelium. This microvilli on the top. This is going to be intestinal lining, things like the small intestine. This allows for more surface area so that we can digest and absorb nutrients through our small intestines. So again, this is simple epithelial, one cell layer thick. Stratified epithelia. Now stratified means that there are multiple layers. Example of this, pseudostratified ciliated columnar epithelial. So they look like columns. They are pseudostratified, which means they have at least a couple layers here. And they are columnar epithelia, which means they look like columns. And Bart Simpson. And these are located in the trachea. This is going to help us get mucus out, so to speak, through the cilia. We have transitional epithelium, and there's multiple layers here. We'll take a look at this, and this is going to be in the urinary bladder. The reason that we have multiple layers here is the urinary bladder kind of stretches. Example of this is this right here is an empty bladder, which we have a thicker lining, and this right here is a full bladder where it kind of stretches it out. And then we have stratified squamous epithelium, again multiple layers thick, and squamous would be, it looks kind of like an egg. And an example of this is going to be surface of the tongue, inside the mouth, mucous membranes. If you've ever cut your tongue or cut your inside of your mouth, it heals very, very quickly. The reason it does is because it has a lot of blank cells that are easily going to repair itself and move up into the right position here. Alright, clinical note, cellular adaptation. We have atrophy, hypertrophy, hyperplasia, and metaplasia. And these are just terms that you need to be aware of. Atrophy, which means decrease in their size. So if you break your arm and they put it in the cast, whenever you get it out of the cast, your arm's going to be smaller. And the reason it's going to be smaller is because it has atrophy. Hypertrophy, increase in size. If you have hypertension for life, your heart will become larger. This is called cardiac hypertrophy. So the heart becoming larger is not always a good thing. There's only so much real estate in there. Hypertrophy, increasing in size. Hyperplasia, increasing in their number. So the actual number of the cells increase and grow. Hyperplasia. Metaplasia, replacement with an immature cell. A metaplastic cell would be something got damaged. We need a replacement for it. It spits out a cell, but the cell may be immature or not be able to handle the job stress that's put upon it. Glandular epithelia. Many epithelia contain gland cells that produce exocrine or endocrine secretions. Exocrine secretions are produced by exocrine glands, which discharge their products through a duct. Example of this is going to be sweat glands, would be considered exocrine glands. Based on their structure, exocrine glands can be categorized, as unicellular glands called goblet cells or multicellular glands. So we can categorize exocrine glands as two types. Either they are unicellular or multicellular. Unicellular are called goblet cells. Mode of secretion. We have three modes of secretion here. We have merocrine, apocrine, and holocrine secretion. Merocrine, the product is released from the secretory vessels by exocytosis. So this is going to be from our Golgi apparatus, and exocytosis is going to occur on the surface of the skin. Apocrine secretions involves the loss of both cytoplasm and the secretory product, and I got pictures of this on the next slide. And then a holocrine secretion. The entire cell becomes packed with secretions and then bursts apart and dies. Fortunately, we have the ability to replace that cell out. Alright, and examples. Merocrine, exocytosis occurs and it expels from the surface of the skin tissue. Examples of merocrine secretion are going to be salivary glands. So you smell a big steak, your mouth starts watering, saliva is secreted from these merocrine glands. Next one on our list is apocrine, and then we lose a little bit of the cytoplasm that gets ready to cause secretion, and the cytoplasm kind of breaks down. Golgi apparatus filling this entire area until it gets the secretion that it needs, and then a section of this will break down, and then it will grow again in that area. And last but not least is going to be holocrine. Holocrine, an example of this is going to be a sebaceous gland from the hair follicle. Builds up, builds up, builds up until the cytoplasm and its contents burst, destroying the cell, and the cell releases its material. Fortunately, we are producing things all the time from mitosis and stem cells down here in the lower, this is a blank cell. By the time that it works its way to the top, and this is a simple or stratified presentation, the answer is it is stratified. By the time that it makes its way to the top, it now becomes one of the cells in a mature cell of holocrine secretion, sebaceous gland sweat. Clinical note here, exfoliative cytology, and there's two things that come up in as far as this clinical note, one of them being a pap test or a pap smear, and this is a screening test used to detect precancerous and cancerous process in the endocervical canal. In the front or the opening of the cervix, what they do in a pap test is they'll scrape off some tissue and they'll do something called a wet prep on it and look for cellular change or cells that look abnormal. They also will send this off to a histologist that will take a look at it. So if you have cervical cancer, there would be a change in the cellular environment. And the next thing that we talk about as far as this is amniocentesis. Medical procedure used as a prenatal diagnosis of chromosomal abnormalities and fetal infections, in which a small amount of amniotic fluid, which contains fetal tissues, is sampled from the amnion or the amniotic sac surrounding a developing fetus, and the fetal DNA is examined for genetic abnormalities. If there was a possibility of an extra chromosome in the genetic makeup or a missing chromosome in the genetic makeup, this can cause severe diseases. Extra chromosome would be an example of Down syndrome. The amniocentesis would probably find the extra chromosome before the fetus would come to full term, and a educated decision could be made over the outcome of the fetus at that point. Whichever take that you have on this, two clinical notes on exfoliative cytology are going to be a pap smear and amniocentesis. And this is going to end section 1. If you have any questions over this, please feel free to call 405-219-7613, or you can write me an email, smithr@imsa.net. Thank you.